The AcidFast staining procedure. The AcidFast staining procedure stains bacteria with a specific cell wall. The cell wall is made of peptidoglycan and mycolic acids. Mycolic acids are lipids, long fatty acid chains that are very waxy in consistency, so therefore they are hydrophobic. AcidFast bacteria stain poorly with a gram stain because the gram stains, the stains that are used in the gram staining, are water soluble. We definitely need a lipid-soluble stain for the AcidFast staining procedure, like carbofuchsin. AcidFast staining can be a diagnostic tool. It identifies bacteria like Mycobacterium tuberculosis, Mycobacterium leprae that causes leprosy, and Nocardia species. So it can be used just like the gram stain as a diagnostic tool because it identifies bacteria that have mycolic acids in their cell walls. AcidFast bacteria that are positive are red, and those that are AcidFast negative are blue. They will be red because they have mycolic acids that will absorb the carbofuchsin stain, and they will be negative. If they are negative, they will be blue due to the methylene blue stain that is used during the AcidFast staining procedure. There are two methods. We will use the heat method called the Ziehl-Neelsen. There's also a non-heat method called the Quinone method, but we will use the heat method in this lab video. Both are named after the scientists that perfected and developed these two methods of identifying AcidFast bacteria. So here's an introduction for you. First thing we want to talk about is the AcidFast cell wall. The AcidFast cell wall, as you can see here, has a peptidoglycan layer, not too thick, and mycolic acids, these waxes, these lipids. And these are the ones that stain directly with the special stain, carbofuchsin, that is used in the AcidFast staining. Now, if we want to compare the AcidFast cell wall to the gram-negative cell wall, you will see the thin peptidoglycan, the two layers, the outer and the inner membrane, and the lipid A that's embedded in the outer membrane. If we want to look at the gram-positive cell wall, there's a thick peptidoglycan, of course the cell membrane underneath, but there's no lipid A, and of course there's no mycolic acids in either the positive, gram-positive, or gram-negative cell wall. So back to our AcidFast cell wall, there's the thin peptidoglycan, and there's the mycolic acid that we will target with our staining procedure today. And again, this mycolic acid is very waxy. It is a lipid, a fatty acid compound. So that's a little bit of an introduction on the AcidFast cell wall. We will move on to the materials. First, of course, the PPE, the baccinerator. We will be using certain bacteria, two bacteria. We have a Mycobacterium smegmatis in liquid broth, and we have a Staph aureus. I don't know if you can see this up close. There's some slimy bacterial culture on the auger slant. So Staph aureus will be on an auger slant for us. Other items that we will need carbofuchsin, which is the primary stain, and this is the one that's lipid soluble. It's nicely absorbed by the AcidFast positive bacteria that have the mycolic acids because it's, again, lipid soluble, so the lipids pick it up nicely. We will use acid alcohol. Acid alcohol contains hydrochloric acid and ethanol. We do not use directly ethanol because we don't want to degrade the lipids that are in the cell membrane of the AcidFast positive bacterial cells. So we use acid alcohol in order to preserve them and not dilute them, but again decolorize the cells that are not AcidFast positive. So the acid alcohol retains the lipids and allows for the staining procedure to continue. We will use methylene blue. This is the counter stain. And any bacteria that will not be AcidFast positive, that don't have mycolic acids, will pick up the blue stain and will decolorize with the acid alcohol, whereas the AcidFast Positive will pick up the carbofuchsin and will not decolorize. We also will use some distilled water for our washings during the staining procedure. We have our nice Tupperware where we can collect all the stains during the staining procedure. We also have bibulous paper that we will use to dry our slides after the staining. We place them between these pages and dry them. We will need optical lens wipe paper. We will need the lens cleaner. And the reason why we need those two items, the wipes and the cleaner, is because we will be using 100x lens with immersion oil. So we will need to clean our immersion oil. Of course, need slides. These are microscope slides. The regular slides, they are glass slides. And here's one of those slides right here. We will need 3 of them. So we're going to have 3 slides during this experiment. And we also will need some cover slips. And these cover slips are placed on top of the slides to prevent any contamination of our lens. There are the 3 slides. We will be labeling the slides first. One will be a staph, the other one will be a mycobacterium, and the other will be a mix of both staph and mycobacterium. So we want to make sure we are going to label these properly, and we want to make sure our slides are clean. Any dirt or any kind of oil will interfere with the staining procedure. So we will take them over here and we will start labeling them. We label with a permanent marker. So we will label the first one Mycobacterium smegmatis. Our next one Staph aureus, Staphylococcus aureus, and the next one will be a mix of the 2 bacteria. Now, one of these will be AcidFast positive and one will be AcidFast negative. Here's the way to label Mycobacterium on the left side, or you can do it on the right side, but you do not want to be in the middle because the ink from the marker will interfere with your staining and your microscopic viewing of the specimen. We will be placing this Mycobacterium smegmatis and making a smear onto this slide labeled Mycobacterium. The next slide, again, we want to stay to the left side, is our Staph aureus slide. We want to make sure we don't go in the middle and interfere with the staining and the microscope view. We will be placing Staph aureus on our slide. Notice the staph is growing on a solid, whereas the Mycobacterium was growing on a liquid. So we're going to have to add a drop of water when we make our smears with that solid bacterium. And here's the mix. Again, you stay on the left side, you don't label in the middle. We will be placing and smearing both staph and Mycobacterium on that slide. So the first thing we're going to do is do our smears, air dry, heat fix next, stain over the steam. You can see the steam here. There's some steaming water on a hot plate. We want to be careful not to touch the hot plate or have the cord entangled anywhere or close to anything that can burn the cord. So we want to make sure we stain over the steam. This is the heat method for the AcidFast staining procedure. We will continue staining and dry our slides and then view them under the microscope. So these are the main steps of the lab. So first, we will do our smears. So smears means to place the specimen onto the slide. We will pick up a loop. We have sterilized our loop, of course. And the first bacterium we will pick up is the Mycobacterium smegmatis. We will prepare the smear by taking a sample from the bottom of the tube. We want a good sample of Mycobacterium smegmatis. And you can see it here. It's kind of clumpy a little bit. And Mycobacterium, by the way, does tend to clump on slides. So it's nice if we can spread it as well as we can in a quarter-sized smear. So we're going to try our best here trying to break up the clumps. We want to kind of spread out those bacterial cells of Mycobacterium so we can view them under the microscope. So we're going to do the best we can to break up the clumping Mycobacterium. But usually we do see the clumping under the microscope. So that is the one smear. You can see that right there. So now we will move on to our next smear. We're going to make our smear of Staph aureus. Of course, we're going to sterilize our loop in between to prevent contamination. Now with the staph, if you remember correctly, the staph was growing on a slant. So because it's growing on a slant and it's very slimy and condensed, it's going to create a very thick smear. So we want to take our loop and first put a drop of water and then smear the staph into the slide. And you can see here it's on a slant, so it's going to be a very concentrated smear if we put it directly onto the slide. So for this reason we will use a drop of water. We put a small little drop of water. Oops, a little too much there, but it's okay. It'll take longer to air dry. We will take the cap off, and usually we have to hold on to the cap, but for now I will put it on the table so I can demonstrate with the slide in my hand. We will pick up some of the staph from the slant gently. We don't want to pick up the gel. Let me show you here. Go really into the slimy bacterial community there. Pick up some from the surface, avoiding to pick up agar. We don't want to pick up agar because that's going to be a big clump in our slide. We cap our staph and then we smear our bacteria staph into the drop of water, into a quarter-sized, if you can see here, smear. We do the best we can. We don't want it too concentrated because it will be too thick to view under the microscope. We want to see individual staph cells. So that is the second smear that we've prepared. We burn our loop, sterilize it, and now we are ready for the mix. Now for the mix, because we will use again the drop of water, we still have that staph and we do have the mycobacterium that's a little clumpy, so maybe this will help in diluting and spreading the bacteria a little bit better. We wait for our loop to cool and we first pick up our staph. Again, we go into the tube. You can see some of the slime there. I don't know if you can see it. There we go. And now we will place and smear the staph into the water, into the drop of water. Make a nice smear, quarter-sized. Sterilize our loop. We are almost done with the smears stage. We will now pick up another loop that's cooler. So we will pick up our Mycobacterium smegmatis and we will take a sample again from the bottom and we will go ahead and smear into the drop of water and the staph that we've added and make our mix. So this now will contain both the staph and the Mycobacterium specimens. Make a quarter-sized smear. Smearing is very important here, so we have to make sure we have good smears. We sterilize our loop. We're going to let them air dry for about 10-15 minutes, and then we will be back to continue our staining procedure. So we will let them air dry for about 10 to 15 minutes. We are back. 10-15 minutes have gone by. We have placed our slides onto the rack and we will follow the protocol in your lab manual. We are following the heat method. You can see that on the upper left. We will have to add some washings, some acid alcohol, and then the methylene blue stage along with more washings. Here's our hot plate. Turn my iPad so you can see that. We have steaming water, so we'd like to place these slides now above the steam. So an easy way for us to, we forgot to heat fix, so I'm glad we're going to do that. We will pick up the slides. You can see they're nice smears after air drying. There's another one that's air dried. And our mix has air dried very nicely. But we need to heat fix. We cannot forget that stage because this will really help fix our bacteria onto the slide. So we light our flame, and with a clothespin, very carefully, we pass the slide over the flame for about 3-4 times to make sure that we heat fix the specimens. Now we can move on to the next slide. Pass it over the flame once, twice. Now we don't want to heat fix them too long because we also have those mycolic acids, so we don't want to melt the mycolic acids. We want to preserve them to the best that we can, and that's why we're using steaming water for the moisture, mainly in the steam. And now we will put aside our flame to be safe, and we will use clothespins to clip our slides on both ends so we can set them on the beaker here with the steaming water. You can see this is a nice, easy way to do this. And usually with a big beaker like this one, we can fit all 3 slides onto the beaker. We clip the next slide, both ends, and we place it on the beaker, making sure we balance them. If they're not balanced, they'll fall into the water, and then the hot water will kill your specimen and will wash it off pretty much. So now we will stain with carbofucin over steam for 5 minutes. But we need to put a little piece of paper over the specimens to make sure that they're not evaporated. I'm going to go get my scissors because I forgot my scissors. And I'm going to cut some pieces of bibless paper, just perfect to fit over the 3 slides that are sitting over the steam. Again, the bibless paper will help prevent the evaporation and the drying up of the specimen. Now, I'm going to do this as fast as I can. I'm going to place it right there, right above it. We have another one, a little too long. Cut it a little bit. Place it right there. Okay. And then we have our third little bibless piece and put it right there. These specimens with the carbofucin literally drench the paper so it will pass right through and nicely be absorbed by all the bacteria that are present on the slide. So all of these bacteria, especially the ones that are going to be acid-fast positive, are going to absorb the stain nicely. This is a lipid soluble stain. So we want to make sure we really flood the slides with carbofucin, and we watch the slides for 5 minutes and literally keep adding carbofucin so the carbofucin is never evaporated. So nothing ever dries out. So we got to keep adding carbofucin and be watching very carefully the slides for about 5 minutes. After 5 minutes, we are ready to continue our staining procedure. The carbofucin has kind of evaporated, so I don't want my specimens to dry up. So I'm going to flood them again. Lots of good carbofucin here. Okay. And now we have to do this for 5 minutes. Keep watching them. 5 minutes is over. We are back. We have taken our slides off the steam, and this is our last slide that we're taking off the steam. We unclip it. And then that little piece of bibless paper that we used, we pick that up with a little tweezer carefully from the end, and we just throw it in the hazard trash. Even though the bacteria now are dead with the stains, they're not harmful. So now we are ready, after we cool our slides for about a minute or so, we are ready to do our washing. We use distilled water for our washings. Now remember, we always pick up the slide from the side of the label and we cover the label with our finger so we don't wash out the label. We spray the water carefully, let it run down the slide, and try to wash as best as we can all that stain off, and sometimes on the back of the slide as well. So this slide looks pretty good. We still have the specimen on there. We haven't lost anything, so that's good news. A little extra washing. Put it back on the staining rack. Pick up the next one. Be careful not to wash off the label. Wash the slide very well, front and back. Watching the specimen. We don't want to wash off any specimen. And our third slide. Careful washing. All the stain, front and back of our slide, and we lay them on the staining rack. The next step, according to the protocol in your lab manual, will be to add the acid alcohol. Now remember, the acid alcohol is a decolorizer. It will decolorize the cells that are not AcidFast positive. The ones that are will retain the carbofuchsin because they have those lipids, and the carbofuchsin will kind of mix in with the lipids. So here's the acid alcohol. We will be decolorizing all our slides, but again, the AcidFast will not decolorize. The non-AcidFast will fully decolorize. We are ready to add the acid alcohol. We let it drip down quickly, few seconds, until we see some of the red come off. And that was enough for that one. You can see here the stain really comes off of that one. So that's the one that's losing the carbofuchsin. And here's the third one. Some of those cells are losing the carbofuchsin with the decolorization step. We are now ready to do another washing. We wash very carefully, front and sometimes back. Place our slide on the staining rack. Second slide, washing front and back. Third slide, washing front and back. Perfect. So now we are ready for the last step of the AcidFast stain, the methylene blue. Now remember, the methylene blue will stain only those bacteria that are AcidFast negative. The ones that are AcidFast positive have absorbed the carbofuchsin and will remain red. We flood the slides with methylene blue very generously. Just flood the whole specimen. And then we allow the methylene blue to sit for 2 minutes. 2 minutes have gone by. We are now ready for the final step before we dry our slides with a bibless paper. We take our water and we wash one last time. Again, bacteria that are AcidFast positive will retain the carbofuchsin. Those that are not have been decolorized and will pick up the methylene blue stain. You can see here we have finished the last slide. We have washed it with water. And now all 3 slides have gone through the staining AcidFast procedure. Put these slides between 2 bibulous pieces of paper and pat them dry. Sometimes they're still wet and you may need to put them between 2 more pages of bibulous paper, like in this case. And we pat them dry with a bibulous paper. We are now ready to place cover slips on our slides and place them under the microscope. Slides will be viewed under 100x. Students will be provided with photos for their lab report. Please remember to clean the immersion oil lens and to clean your lab area.